Hey everyone, in this video we'll be discussing about Gram staining, which is a type of differential staining. So differential staining technique is a process which uses more than one chemical stain, and using multiple stains can better differentiate the microorganism based on their structure and cellular components. So different types of differential staining technique include Gram stain, acid fuzz staining, endosperm staining, capsule staining, and flagellar staining. So Gram staining was developed in 1884 by Hans Christian Gram. It is one of the most widely used procedure to distinguish between gram-positive and gram-negative bacteria. So in the first step to Gram staining technique, we have to prepare a smear using distilled water, and to it we add crystal violet stain, which is the primary stain. It is a basic dye, and this we have to keep it for 1 minute. Followed by rinsing with water, we add iodine, which acts as a mordant. It helps bind the first dye, that is crystal violet, more strongly to the membrane of the bacteria. We add the iodine solution, keep it for 1 minute, and then we rinse it with water. So in the third step, we use 95% ethyl alcohol. We keep it for 10 to 30 seconds, and then we rinse it with water. In this step, the alcohol acts as a decolorizer. In the fourth step, we add safranin, which is a counter stain. So this is also another basic dye. We keep it for 45 seconds and then we rinse it with water and blot dry. So these are the steps we discussed right now. In the first step, we add crystal violet stain, and in this stage the cells stain purple. In the second step, we add iodine, the cells remain purple. In the third step, we add alcohol, which acts as a decolorizer. In this stage, the gram-positive cells remain purple, while the gram-negative stains become colorless. And in the fourth step, we add safranin as a counter stain. And in this step, the bacteria, in case of bacteria, the gram-positive one remains purple, while the gram-negative bacteria, they appear pink or reddish, like saffron, the color of safranin. So when we view under the microscope, we can see that the gram-negative bacteria appear pink in color, and the gram-positive bacteria appear violet or purple in color. Hey, so why did the gram-positive bacteria remain purple color while the gram-negative bacteria appeared in pink color? The reason is because of the difference in their outer membrane. Gram-positive bacteria has a very thick peptidoglycan layer, while the peptidoglycan layer in gram-negative bacteria is very thin. And the membrane of gram-negative bacteria has a lot of lipid in its membrane, in its outer membrane. So during the dehydration step, using alcohol, the membrane gets dehydrated and the crystal violet iodine complex formed during the initial stages of Gram staining technique will leak out of the membrane of the gram-negative bacteria, making it colorless. And when we stain with safranin, which is the counter stain, the gram-negative bacteria accept the stain, while in case of gram-positive bacteria, there isn't much lipid-rich membrane, and hence it remains purple in color as the peptidoglycan layer retains the crystal violet iodine complex in a strong manner. So that is the reason why gram-positive appear purple or violet in color, while the gram-negative bacteria appear pink in color. So thank you for watching the video. Please do subscribe and keep watching.